Wajia, everybody. My name is Natasha Louttit. I work in the Eeyou Marine Department of the Cree Trappers Association. I'm the Wildlife Liaison Officer. Here at the Cree Trappers Association, our mandate is to work closely with the Eeyou Marine Region Wildlife Board to facilitate research projects in order for us to effectively manage wildlife. Although polar bears are not new to the area, there has been very little to no research done, so we had a need to gather scientific data on the conditions of the polar bear population in southeastern James Bay, which we refer to as the Cree Eeyou Marine Zone. That's where Alex comes in with her research project into polar bear ecology and the start of our collaboration with the Eeyou Marine Wildlife Board and McGill University. This research project will help us answer the questions we have about the health, condition, diet, abundance, and genetic analysis of our polar bears along the eastern coast of James Bay from Waskaganish up to Chesapeake. Traditionally, Cree do not harvest or trap polar bears for sustenance or their fur, so we don't look at them in terms of a food source, but they do require responsible management since they are a federally recognized species at risk. The polar bears in EUSG may also be a genetically unique population. In the past few years, climate change has made itself more visible. Our land users have reported clear and distinct changes to regular weather patterns, that our winters are getting shorter and milder. Ice does not freeze as it used to, which changes the safety conditions for traditional hunting practices during the winter. And in some areas, there is open water all year round. As part of our mandate and services the CTA provides, our CTA EMR local officers in each of our coastal communities collect all polar bear sighting and observations reported by our land users and community members. An unusually early spring opened what ice remained in the bay so fast that it quickly forced polar bears to offshore islands and along the coast during our annual spring harvesting of geese, and most of our community members and their families had already moved out to their spring hunting camps. It's clear to us from information that we've gathered from observation reports coupled with climate change that encounters with polar bears is rising at a very fast rate. That is why it's so important that the Cree Trappers Association invest in research to determine the status of our polar bear population in order to establish effective management and conservation guidelines. These tools will promote healthy wildlife management and, most importantly, help us educate ourselves so that we can provide and establish services such as proper polar bear deterrent methods and safety measures to ensure that our land users will always remain safe while continuing to keep traditional activities and practices alive and well. Miigwech.
Hi, I'm Alexandra Langwieder. I'm a master's student working with the Eeyou Marine Region Wildlife Board, Cree Trappers Association, and I'm supervised by Murray Humphreys at McGill University. Our objectives were to launch a field-based polar bear study, and through this, investigate the genetics, diet, body condition, and habitat use of the polar bears in the Eeyou Marine Region of James Bay. To do this, we are using camera traps, hair snares, and Cree knowledge interviews. Camera traps provide us with photo and detection data and help us understand polar bear body condition and habitat use. Hair snares provide us with hair samples containing DNA and stable isotope signatures that inform us on James Bay polar bear population genetics, individual movements through the study area, and diet. Cree knowledge interviews contribute Cree knowledge of polar bears in the Eeyou Marine region and inform us on polar bear habitat use, historic and current trends in polar bear movement, and local polar bear behavior. In summer 2021, myself and these field teams from Waskaganish, Eastmain, Wimminji, and Chesapeake deployed 37 hair snare and camera trap sampling stations across 400 kilometers of eastern James Bay. I would now like to introduce George, Henry, and Ernie, part of the field team from Wimminji, who will explain some of the methods we used and the places we went.
We set up a snare. We use steel posts, put them in a triangle shape with barbed wire around it to collect the fur, and we put bait in the center.
The camera was set up just to catch what predator crosses the barbed wire. Sometimes it's not a polar bear, it's a caribou, or we even encountered a black bear, and possibly other species, fowl, birds, geese.
As the bear crosses the barbed wire to investigate the bait, hair gets caught in the wire and we come back later to collect it. The camera allows us to link the hair sample we collect to the photo of the bear that left it there, which is illustrated here. On the left, we have the bear leaving the sample, and on the right, I'm collecting that sample. With this, we're hoping to be able to link diet in the spring, when the hair was growing, to the body condition of that bear during the summer.
When we go to one of these traps, we usually look around for polar bear. We had one guy walking around, make sure it's safe. But, we check all the hair sample. We walk around, we check all there's hair samples on the wire, and if there's, then we collect them. Then we mark on what date and what trap number is this. And we check the camera to make sure it's not on the ground, and we set it up again. And we after that, when we're all done, we rebait the bait. Are there lots of polar bear out there? That's mostly the question I ask from the people. Are there lots of them? Are they dangerous?
A lot of the people ask, how long does it take you to get out on those islands? And man, it's never the same answer. I'd say it depends on the weather, depends on the tide. And they ask, are they coming to shore? Are they gonna enter the cabins, or so on?
James Bay is a tricky bay. It can come up anytime. It's so shallow, but it comes up to 8 feet, 6 feet sometimes. And the unknown shoals is mostly uncharted water. And it's dangerous when you go on uncharted water. You don't know what's there. And then plus, there's a lot of currents. It's like a river out there.
He's going to tell a story of that trip we did.
We that's collecting the cameras, the equipment that was out there. We were out there 12 hours on the water. It started early, it was calm in the morning. The wind picked up a little bit, then it kind of died down. We ran to Twin Islands, South Twin, North Twin, Walter, came back to Goose Island, to Solomon Temple Islands, and Pebble Island. It was a busy day, but it was still daylight when we got back. We managed to do all that.
We detected polar bears at 13 of the 37 sampling stations, mostly on offshore islands. With these stations, we gathered 133 polar bear observations from the camera traps and 117 polar bear hair samples from the hair snares. The hair samples are still being analyzed, but we have some preliminary results to share from the camera trap observations. We assessed each bear using the standardized polar bear fatness index, which scores bears from skinny to very fat. We found that most of the bears we detected were in average body condition, with some being fat and a few being thin. The black bar on the left shows the number of unclassified bears, which were unclassifiable because a previous polar bear had knocked the camera off the post, and I was just able to detect a foot or a nose investigating the camera. Our next steps are to prepare for the Cree Knowledge interviews and winter fieldwork. We will be deploying the sampling stations again in February and March of 2022 with the teams from this summer to determine if the same polar bears remain in James Bay all year and what their diet and habitat use is at this time.
I'm curious of what are the results of the study. If the same bear that was in Pebble Island can be on the other islands, like south of Old Factory or even North Twin, or the one that's hanging around at Walter is the same bear that we caught the samples off on North or South Twin. And I'm very curious of what other stuff they eat, what's their I'm sure it's not only seal, I'm sure it's other stuff.